Well, welcome to Notes from the Lab. This is Dr. Denise Fautsman at the Mass General Hospital and Harvard Med School, and we're doing a series of podcasts to get everybody up to speed on the newest data on the BCG vaccine. And we're very honored today to have one of our best scientific colleagues, Dr. Christine Benn from the University of Southern Denmark, here to come and give us her updates on what's happening globally on the BCG vaccine and the non-specific effects of the BCG vaccine. So it's my honor to introduce you today, and I'd love to start out by you just telling us a little bit about all your excitement about the non-specific effects of the BCG vaccine.
Well, that's very easy because I am truly excited and I'm getting more excited by the day because we have so promising evidence for this vaccine's broader non-specific effects against not least infectious diseases, but also the more we learn against all kinds of diseases. And yeah, I don't know where to start. It's a 100-year-old vaccine, but I guess the listeners to your program will already know that.
Well,
It
not
was
necessarily. We think a lot of
okay.
lay people will also. So tell us a little bit about BCG because in the United States, remember, everybody thinks it's Boston Consulting Group still. So it's always good to get a little background on this amazing vaccine.
Okay, so let me tell your listeners that this vaccine was invented by Calmette et Guerin, who were two Frenchmen, and it's called Bacillus Calmette et Guerin after the two inventors. It was their attempt to make a vaccine against tuberculosis in the 1910s that led to the development of BCG in 1922, and they were looking into lookalikes of the Mycobacterium tuberculosis that caused tuberculosis in human beings, and they found the bovine Mycobacterium bovis, and they thought if we inject that in, or actually they started out giving it orally, but if we modify it and make it a bit weaker, and then we give it to human beings, that might confer some cross protection against tuberculosis. And that indeed turned out to be the case, that this vaccine could protect pretty mediocrely, not 100% against tuberculosis, but it had some protective effects against this disease, for which there wasn't any really good cure and which still killed a lot of people at the time the vaccine was developed, and actually still kills people in low and middle income settings. So they developed the vaccine, they started using it, and actually Calmette, he publishes in 1931, pretty unnoticed. It at least never really caught fire at that time, but he published that when they started using BCG in Paris among children at high risk for tuberculosis, in the control group, it was normal that 20 to 25% of the children could die over a short observation period. But in the group that received BCG, it was down to 4 to 5% who died. So he was noting at that time that reduction in all-cause mortality seemed to be far bigger than it could be anticipated just based on the protection of the vaccine against tuberculosis. It maybe conferred some non-specific protection of the children, he actually speculated. And that was followed up by reports from many other countries that started using BCG of quite dramatic reductions in all-cause mortality. For instance, there was a Swedish pediatrician who rolled out the BCG vaccine in northern Sweden and discovered that children died much less when they received the BCG vaccine, and particularly also in the neonatal period in the first month of life, where children don't normally die from tuberculosis. There he also saw reductions in all-cause mortality. So also he speculated that BCG could confer some kind of non-specific protection of the children. But I think it's actually a bit sad testimony to how medical science develops, because nobody picked up on this, and it kind of was forgotten for decades before our group starts working with BCG or starts looking at all kind of routine vaccines in Africa, in Guinea-Bissau, a small West African country, and rediscover, so to speak, that BCG vaccine is associated with tremendous reductions in all-cause child mortality that cannot at all be explained by the prevention of tuberculosis.
I mean, that's a study that if somebody mentions your name, I think of. So do you want to tell everybody what was the goal of the study and what you found?
Well, initially, we actually found that measles vaccine conferred very strong beneficial effects, and that led us to and repeated that observation. So that led to the hypothesis that measles vaccine could induce non-specific protection against the diseases that caused high mortality in West Africa, and that was primarily infectious diseases in children. So discovering that led us to investigate, or not me personally, I should say, that was my mentor, Peter Arby, at that time, who started out investigating all vaccines. I joined him only in 92 for that inquiry. But looking at all the other vaccines, it turned out that BCG seemed to have that same beneficial effect as measles vaccine had. Initially, it was just observational studies, so comparing children who were vaccinated with those who weren't vaccinated yet. And there could be reasons for there could be other differences between those vaccinated early versus later that could explain why they would have different mortality. For measles vaccine, we could exclude that this was purely differences between the groups because we delivered the measles vaccine and we knew the factors that differed between those vaccinated and non-vaccinated. For the BCG vaccine, there was this interest in actually trying to substantiate how big was this effect and could part of it be explained. It was a very big effect, so it seemed implausible that that should be explained by inherent differences in those who chose to be vaccinated and those who didn't. But nonetheless, it was really what we were really happy with was that there was an opportunity to do randomized controlled trials with BCG. And that opportunity was available because at the time when we did our study in the zeros, the 2000s in Guinea-Bissau, that it was policy not to vaccinate children with low birth weight immediately at birth. So BCG was recommended at that time to all neonates.
I didn't know that fact. I didn't know that fact.
Yeah. So that was only for normal birth weight children. So low birth weight children were exempted from BCG vaccine at birth, and it was postponed until they reached 6 weeks of age when they came for their first routine diphtheria, tetanus, pertussis vaccine. So we could, with our observational data in hand, which suggested that it was actually really beneficial also for the children with lowest birth weight to get BCG at birth, we could get ethics permission to randomize children with low birth weight to BCG or the usual treatment, which was delayed BCG. And that provided a window of opportunity or a window of observation in the neonatal period in the first month of life, where we could compare those who were randomized to BCG with those who were randomized to no BCG, and confirm that a large chunk of the beneficial effect of BCG was in fact due to non-specific effects. It reduced the risk of dying from sepsis and other other infectious diseases such as respiratory infections in the first month of life, infections that would otherwise have killed these children. So the reduction of BCG in all-cause mortality, and that was confirmed in 2 other trials, was around a third. So with a very high child mortality in Africa, you can imagine that in absolute numbers, we are talking about a possibility of saving several 100,000 child lives every year with BCG vaccine.
That's why it's always hard to give a lecture after you, because you talk about children who die who didn't get it, children who live, and then I get up and I talk about blood sugars. Okay, so it's really hard to give a lecture after you, Christine, just so you have that in context in the big picture. Do you want to go ahead? Yeah.
I just want to mention that actually studying BCG in a context with such a high infectious disease pressure has been like a magnifier to the BCG effect. So I think that is if you are in a context with very high infectious disease pressure, anything that changes your immune system will be seen very clearly and very early on. So that is what we could do in our context, but I think it's extremely important, and I would not undervalue in any way what other people are doing, looking at other parts of human life. And of course, this observation builds on the tragic fact that there is such a high infectious disease mortality in Africa.
Yeah,
but if I
remarkable, yeah.
But if I can jump in history, now we've been back in 1922, we've been in the zeros, and we published our first randomized trial in 2011, and our meta-analysis of 3 trials in 2017. I'm just really happy that at this moment of time where we are talking, just a month ago, there was in BMJ, one of the leading medical journals, a huge trial from India enrolling more than 5,000 children confirming these very strongly beneficial effect of BCG vaccine. And this was in neonates who were even more fragile than those in Guinea-Bissau. They were all weighing less than 2 kilograms and would not normally, again, get BCG in India, according to policy. But they were randomized to BCG or no BCG, followed for a month. And they had almost a halving in their risk of dying from infectious diseases. And in this beautifully conducted trial, they also looked at microbiology, at the actual pathogens, disease organisms that killed the children, and they could show that the effect of BCG was, for instance, on Klebsiella, which is a bacteria that can kill babies in the first month of life, and which is one of the main causes for the antimicrobial resistance that we are seeing almost exploding worldwide. Mm-hmm. So these results are truly encouraging in terms of showing a road forward for decreasing neonatal mortality and actually also at the same time combating antimicrobial resistance. So yeah, exciting times for BCG.
Yeah, so kids are one scene, but obviously adults are a whole nother scene. So do you want to kind of summarize maybe what happened in the COVID pandemic and where the data is now? Because for our listeners, with such strong data that BCG is a platform to protect from many infectious diseases. Of course, when COVID hit, everybody ran out to do their trial. And there were, I think you'll agree, many mistakes. People forgot the populations they were dealing with that already had BCG, or people used different strains of BCG, or people didn't wait enough time. So do you kind of want to summarize where you think that data now stands on adults and COVID and BCG?
Happy to, yeah. And we both remember that we were at a conference, the Optimize meeting in Cambridge in February 2020, yeah. And we almost didn't make it home before the whole world closed down. But indeed, at this conference, which is focused on non-specific effects, we were all very encouraged to go home and start studies and looking at BCG, yeah. Whether that could help us. Using BCG could be a stopgap vaccine for until specific vaccines could be developed. So a lot of protocols were written, a lot of studies were carried out, and that was fantastic times for researchers into BCG too, and very full of a lot of hard work for us all. We personally conducted 3 randomized trials with BCG vaccine in Guinea-Bissau and in Denmark. And in the end, to summarize all the results, a total of 15 trials were carried out, and they showed quite divergent results. So there were studies like your own in type 1 diabetes patients that showed a very strong protective effect of BCG against COVID-19. This was in a context of quite vulnerable patients who also had received, before the pandemic even broke out, numerous BCG vaccines.
Yeah, and exactly.
In them, you saw, if I remember correctly, a 91% reduction in the risk of getting COVID in the first published.
Equivalent to Pfizer and Moderna.
Yeah.
But you're right. We had Christine called me up for the context. He goes, Denise, you already have all these vulnerable people enrolled. And how many vaccines do they have? Why aren't you studying COVID? I go, I'm hurrying as much as I can. So our trials were a little bit a head start.
We had a wonderful head start, yeah.
Yeah.
And there was also a Greek group that had already published just before the pandemic a randomized trial of BCG in quite vulnerable elderly who were discharged from hospital and randomized to BCG or saline and followed for a year. And they could show that BCG reduced the risk of infections, also half the risk of infections in these vulnerable patients over 60. And the effect was particularly strong on respiratory infections. And they also had the setup to very quickly run another trial during COVID, and they also found protective effects. This was in Greece, where BCG has been used in childhood, and where they found also the same beneficial vulnerable patients, where BCG in the COVID period was a second dose of BCG. It wasn't the first dose of BCG, and very protective effects. But other studies, including, and I would have to say that the study that disturbs me most or is most confusing is the study in Dutch patients that were just as vulnerable as the Greek patients, at least on paper. They also were multimorbid. They were in the hospital. They were discharged and they were randomized to BCG or no BCG. And they were not seeing the same benefit from BCG on COVID-19 infection. And there, in fact, I think it was 25%. It should be said that in the Netherlands, BCG was never used, just like in the US. So a large chunk of these patients had never received BCG before, and they received a primary BCG, but 25% of them had received BCG before, and they didn't see any benefit in that group either. So there are places where in my picture, it almost, in my world, it almost adds up to, we can explain the totality of data, and that is obviously what we are always interested in as researchers, to understand how all the data kind of fits into the joint picture. And I wish I could say most of the data suggests that if you got repeated BCG and you were a quite vulnerable person, then you had some protection against COVID-19.
And some
but
time. And some time.
And some time, and you might be right about that's a very valid point, Denise. Yeah, that you might be right that the follow-up time was still too short in the Dutch study after BCG. And I mean, we also did a study in elderly in Denmark and found no effect on COVID-19, but they were super healthy elderly. And I think that's another factor that has been shown now, that BCG can train the innate immune system. And we didn't really touch upon that, but that's at least one of the mechanisms that are in play to explain why we see these very dramatic and quick effects on neonatal mortality. BCG can train the innate immune system, but it only does so if your baseline is actually quite low. So it doesn't really sort of improve. It's hard to improve on a winning model. And I think the elderly that we enrolled in Denmark, for instance, they all, had their probiotic yogurt for breakfast and exercised 5 times a week. And they were so well functioning that they also came out during the pandemic to come and meet us and get enrolled in the randomized trial, etc. They were perhaps hard to improve on. But anyway, to cut a long story short, these 15 trials showed significant results. They have been used by some people to say, well, there you go, there was not really any effect of BCG, which I think is a too hasty conclusion, because there were effects in some studies that were quite dramatic, like in your trial, for instance. And also, what is really intriguing is that if you look across all the trials that reported mortality data, so where one or more of the participants in the randomized trial during COVID died, then it's completely consistent that in the BCG arm, the group of people randomized to BCG in the clinical trial, there were fewer deaths than the control group that was randomized to saline. And over the 6 to 12 months of follow-up, the relatively short, as you've already pointed out, relatively short follow-up period, if we summarize all the data from all the trials, you have half the risk of dying if you receive BCG than if you receive saline. And that is highly statistically significant. So what the data indicates is that the minimum reduction in mortality based on this data is 15%. But the point estimate, as I mentioned, it points towards a halving in mortality. But you can imagine that just if just 15% is correct, then it's a wonderful, wonderful vaccine to prolong survival
and healthy survival. It's not just healthy survival, because the vaccine's so safe.
Yeah. That's also a very important point. Yeah, it's been used for more than 100 years now. We know it's very well characterized in terms of its adverse reactions and adverse events. And I think the only place where you should worry about a BCG vaccine is if you're severely immunocompromised. We know that you shouldn't take the vaccine if you have fulminant AIDS, because in people with extremely almost non-existing immune systems, then BCG can develop into an infection in itself. It can create an infection in itself. But otherwise, yes, a very safe vaccine that you can even give to vulnerable newborns.
Yeah, which is amazing. So do you think any other live vaccine is going to come close to BCG, or is BCG such a strong live vaccine?
I think you're alluding to something which is important, and that is the liveness of BCG, because BCG is indeed the live bacteria that is that is still viable, and that's an important feature in the production, that it's made with viable bacteria. It's sent to market with a certain count, a certain number of viable bacteria, and it shouldn't be used after a period when these bacteria don't survive any longer. And it should be used once dissolved. It should be used. It comes freeze dried, but it's dissolved in saline, and the bacteria comes alive, but they only stay alive for 4 to 6 hours. So the vaccine should be discarded if it's not used within that short period. So it contains the live microorganisms, which is truly fascinating, because that seems to be extremely important for these non-specific beneficial effects. Because in fact, we have seen the same, maybe not to the same magnitude, maybe not of the same duration, but we have seen similar, very strongly beneficial non-specific effects of other live vaccines, like measles vaccine, oral polio vaccine, and also historically the old smallpox vaccine. So it seems that having these live vaccines, which in fact is kind of mimicking having a mild natural infection, does train your immune system, does confer benefits, and teach it important lessons that the immune system can use whenever it's challenged with other pathogens in the future, which makes enormous evolutionary sense. That of course the immune system would love to learn more from meeting
all the time
a measles virus. That it's not just a matter of being prepared to meet another measles virus in the future. It's about being better equipped to meet other pathogens in general. And there I find it important to mention that currently we are pursuing a new observation that we find enormously intriguing, namely that children born of mothers who themselves receive BCG seem to benefit more from getting BCG, which again, evolutionary speaking, would make sense, that you are actually primed to meet from your mother to meet the same pathogens that she has experienced. So you're born with an immune system that is adapted to meet the same pathogens that your mother has met. Yeah.
Do you think BCG goes through breast milk? I get this question all the time.
I don't know. That's a good question.
Okay, I was just wondering, because is it the immunity that's transferred to the mother, or is it actually the microorganisms that are transferred to the infant, and that confers the immunity?
Also, with the maternal priming, we see that even if the mother received BCG as a child, so it's not something that is because she's acutely vaccinated. It's and actually you cannot give BCG in pregnancy. It's all live vaccines are contraindicated if you're pregnant, or mostly, I mean, for theoretical reasons. It's not like there's supposedly. I think it's out of caution. So we don't have any mother who will be breastfeeding and who had BCG.
Who will get a vaccine at the same time. Okay.
No, so I think that's most likely this is epigenetically transferred. And I'm saying that based on studies where we looked at also fathers of children and seemed to see the same kind of priming. So if the father had received BCG,
so that would not be also breast milk. That would not be maternal fetal circulation. Okay.
And also it has been shown, I think, that BCG also does, I mean, in urine studies, there are you can inherit also traits from BCG primed male to the offspring. So I think there is mechanisms or that actually has been shown as a mechanism to transfer that immunity. So through the germ cells.
So one last comment, so we stay on time for our BCG updates. Where do you think the field's going with multiple vaccines and the sequences of vaccines?
So what we have seen is that there are these benefits of live vaccines, which seem to be much stronger and more pronounced and important than the benefits you confer with the non-live vaccines. And that means also that we see in our magnifier in Guinea-Bissau in West Africa that children survive much better if they have a live vaccine as their most recent vaccine than if they have a non-live vaccine as their most recent vaccine. So some of the non-live vaccines can be, you don't want to stop vaccinating against diphtheria and tetanus and pertussis, which are deadly diseases. but actually it looks like the vaccines in themselves do not do any innate immune training. They don't stimulate the immune system beneficially. In fact, we have evidence for the contrary. But fortunately, if you give a live vaccine after a non-live vaccine, that seems to train the immune system beneficially. And analysis have been done which show that based on data that actually came out of a World Health Organization review of the non-specific effects, data from that review put into a model comparing the current vaccination schedule in low and middle income countries with one that was optimized for its non-specific effects, making sure that children have a live vaccine as the most recent vaccine for the largest possible period of childhood, would reduce all-cause child mortality by 1 million deaths per year. So it could make a dramatic difference to use the currently used vaccines, but in a slightly different schedule. And yeah, exactly. With live vaccines as the most recent vaccine for the major part of childhood, that would not, you wouldn't miss any of the specific immunity conferred by these vaccines, but you would optimize the immune training that would make the child
healthier overall.
Yeah, yeah.
Wow,
it's
such exciting times because in theory, most research has traditionally last 20, 30 years. Well, we got this bacteria and we want to make a vaccine against it. And then the next study, well, we got a different virus. We want to and nobody thinks about the bigger picture of interactions or what else does this vaccine do? And usually you don't have the time and resources to look at those other things. But I think your work is so elegantly showing to the world of how these live vaccines are better than we ever expected. And of course, my favorite, I think your favorite too, is still BCG. So we'll have more to follow and share. So thank you for being with us today. It's so appreciated.
Thank you for inviting me.
Okay.